Okay, so hello everyone. I hope you're well wherever you're watching this from. So the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. The topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support your career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. So please see the website for more details. So I'm Carl, I'm a research fellow in Julie Aston's group at the University of Leeds, and I'm delighted to host today's webinar, which is titled Insights into the Role of Ribosomal Proteins in Development, and is part of our dedicated early career research program of webinars. So today we are delighted to have Katy Grobicki and Shajahan Anver with us, who are going to share their work with you, the molecular biosciences community. So before I give the stage over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type it in the question box as shown in the image on the screen, stating who your question is for, and we will try to answer as many as possible at the end. So our first invited speaker today is Katy Grobicki from the University of Cambridge. Katy is a fourth-year PhD student in Philippe Karam Teixeira's group at the University of Cambridge. With an interest in the translational regulation of development, Katy has been using Drosophila to investigate the roles of ribosomal protein paralogues in the germline, and will be sharing some of her recent work with us today. So over to you.
Okay, and hopefully, Carl, can you see my slides okay?
Perfect.
And great, so hi, I'm Katy, as we said, and yeah, thank you very much to the organisers for allowing me to share some of my PhD research with you guys today. Sorry, okay, it's just a bit laggy. So we all know that ribosomes are essential for all translation in all cell types, and that the ribosome is made up of 79 proteins and 4 ribosomal RNAs. And I'm mostly going to be focusing on these ribosomal proteins today. So I work in Drosophila, and in Drosophila, you guys can see that now. So in Drosophila, any mutations or loss of any ribosomal protein usually causes this classic minute phenotype, which includes very slow larval development and short, thin bristles. And this was always assumed to be due to too few ribosomes being assembled, and this being insufficient to support translation and proliferation of particularly fast-growing tissues such as the bristles. Yes, that's better. Okay, in humans, defects in ribosomal proteins or ribosome assembly factors cause developmental diseases known as ribosomopathies. But the interesting thing is that mutations in different ribosomal proteins or at different stages of ribosome biogenesis can actually cause quite different phenotypes by seemingly affecting some tissues, different tissues depending on which ribosomal proteins are mutated. Now this, of course, seems very interesting and rather strange, because the ribosome is required to produce all proteins in all cells. So why would mutating one ribosomal protein affect one tissue more than another? And part of the answer to this could be that ribosomes are actually not identical as we have long assumed them to be. And there's growing evidence in organisms from yeast to humans that ribosomes can actually be quite heterogeneous, and that this heterogeneity can be introduced in a variety of ways, including post-translational modifications on the ribosomal proteins, chemical modifications on the rRNAs, through swapping in and out different ribosomal proteins for their paralogues, or through the whole ribosome associating with different accessory proteins. And factors such as all of these combined could perhaps affect the speed and fidelity of translation elongation, the localization of ribosomes within the cell, and perhaps even the transcript selectivity. And all of this together has led people to hypothesize the existence of specialized ribosomes, which could perhaps exist in specific developmental times or specific tissues and translate certain subsets of mRNA. And so far, there is definitely evidence for ribosome heterogeneity, but in animals at least, there's less solid evidence for whether or not this has a function as specialized ribosomes. So ribosomal protein paralogues are what I'm going to be talking to you about today. So genes get duplicated over time, producing pairs of paralogues, as we know. And due to 2 copies now being present, there will be redundancy. So one of these copies might accumulate mutations, become a pseudogene and be lost over time, or alternatively, it might gain a new function. And in Drosophila, there have been 14 independent duplications in the lineage leading up to Drosophila melanogaster. So there's not been any whole genome duplications. Each of these is a separate event. And melanogaster isn't unusual with these 14 duplications. As you can see, there's actually far more duplicates present in some other species, and humans actually have 19 ribosomal protein paralog pairs as well. So within these pairs of paralogs, one gene is expressed ubiquitously at high levels and it is essential for survival, with homozygous mutation is lethal and heterozygous mutation causes this minute phenotype that we mentioned earlier. We were more interested in the other paralogs, so namely as non-canonical to differentiate between them, as these had very specific expression patterns. They're broadly low expression, but seemed particularly to be enriched in the germline. So much of my PhD has been focusing on understanding whether these non-canonical ribosomal protein paralogs have a function in translation in the germline. So I began the project by aiming to mutate each of the 11 ribosomal protein paralog genes that were specific to Drosophilids. So these range between 2 million and about 70 million years old. And I managed to create between 5 and 8 different knockout alleles for each of those genes. Then validated them via genotyping Western blots where antibodies were available, and then moved on to understanding the phenotype. So first off, I wanted to check whether all of the mutants were able to homozygose or whether this was lethal, but and then also look at these homozygotes and see whether they displayed slow growth, bristle defects, or any of the other features of the minute phenotype. But actually, it turned out that all of the mutants were viable as homozygotes. Under lab conditions, they didn't seem to display any defects or phenotypes, and none of them caused any features of the classic minute phenotype. So then, as most of the genes showed enriched expression in the germline, of course, I wanted to assay fertility. So as you can see, this is only looking at males here. Most of the males there's some variability in offspring number, but all of them are fertile to a degree, which falls within the range of the control. And this was quite surprising because most of these genes show enriched expression in the testes, and this is more frequent than in the ovaries. Meanwhile, moving on to the females, you can again see that most of them are fertile. However, there's one gene, RPS5B, for which the females were completely sterile and never laid any eggs. So after this kind of crude method, I then also dissected ovaries and testes of every genotype to test if there was a more subtle phenotype in any of these other genes, but all of them except the RPS5B ovaries looked wild type. So just to quickly take you through the Drosophila ovary, because that's what I'm going to be showing you quite a lot of pictures of. So each female fly has 2 ovaries, and an ovary is made up of around 16 ovarioles. And an ovariole is, oh, where's it gone? Sorry, it's just not quite loading. Yeah, an ovariole is basically a chain of developing egg chambers. So at the anterior-most tip, you have the germarium, which is where the stem cells will reside. Here in their niche, then the stem cells divide to produce a daughter. This daughter undergoes 4 rounds of cell division, but with incomplete cytokinesis. So this produces a 16-cell cyst of interconnected germ cells, which are sharing their cytoplasm. This cyst is then enveloped by somatic follicle cells to produce a unit that we know as the egg chamber. This egg chamber will then grow and develop, and you can see it becomes much larger in order to finally produce an egg. And of these 16 germ cells, 1 will go on to become the oocyte, and the other 15 become nurse cells, which will provide all of the proteins, RNA, and organelles that the egg will require for embryogenesis. So getting back to RPS5, this is where RPS5 sits in the ribosome. And if we go back to the pair of paralogues, RPS5A, this is the one that's essential for survival. RPS5B is the one that seems to be required for female fertility. Actually, looking at the structures, at the protein sequence, they're over 80% conserved, and almost all of the differences are in this flexible, unstructured N-terminal tail region, which kind of sticks out of the ribosome, so may perhaps be under weaker selection. Then, more interestingly, the 2 genes show differential expression patterns. So RPS5A is highly expressed in most tissues, but you can see that there is more expression in the somatic cells around the outside of the egg chamber compared to in the germline in the center. Meanwhile, RPS5B, you can see it's much more highly enriched in the germline. And that for both of them, these differences seem to start just where the first egg chamber exits the germarium. Sorry, it's just lagging slightly. Okay, so this is what a nice wild-type egg chamber looks like in real life. On the left, each of these kind of fingers is an ovariole, and on the left we'll have our stem cells in the niche. And then as you move from left to right, the egg chambers are maturing to produce the most noticeable feature here, which is these lovely mature eggs. So to compare with the RPS5B mutant, the early stages all look pretty normal, but then at mid-oogenesis, all of these egg chambers are dying, and you can see that mature eggs are never produced. Zooming in a bit closer, you can see that in a wild-type egg chamber, there is a neat monolayered epithelium of the somatic cells surrounding the egg chamber, whereas if we look at the RPS5B mutant, these follicle cells have become very disorganized and multi-layered, and that this is occurring before the germ cell death, so isn't merely a consequence of the egg chamber like collapsing as the germline dies. So as there was both a germline phenotype and a somatic phenotype, I wanted to explore where RPS5B expression is actually required. So using FRT clones briefly to test this, I could see that when RPS5B is only mutated in the germline, it reproduces the phenotype that we see on the left, whereas when RPS5B is only mutated in the soma, it basically has no effect. Doesn't cause any multi-layering either. So RPS5B expression only seems to be required in the germline, and this somatic multi-layering phenotype is a result of downstream signaling from the germline to the soma, which must be disrupted in the mutant. So then to further understand what's actually going on in these ovaries, I looked at a variety of different pathways involved in growth and development. One of the key ones obviously being TOR. So TOR pathway is very highly conserved and basically regulates growth in response to nutrition and a variety of different stresses. And it TORC1 particularly is known to control ribosome biogenesis and translation. So I wondered if it might be altered at all, its activity in my ribosomal protein mutant. So I've used an antibody against phosphorylated RPS6 basically as a readout for this branch of TORC1 activity. If we look at the heterozygote, you can see that phosphorus-6 is only seen in these early germline cysts, so it's absent in the stem cells and then absent again in these later stages of oogenesis. And this recapitulates what we've seen in other wild-type flies. Meanwhile, in the RPS5B mutant, you can see that there's strong ectopic activation of TORC1. The phosphorus-6 basically begins as soon as the first egg chamber leaves the germarium and carries on all the way up until these egg chambers die. Interestingly, there is also phosphorus-6 activation in the somatic cells, including in yellow here, we've got the follicle stem cells. And you can see that the follicle stem cell activation is only seen ever in the mutant. And I hypothesized that some of this TOR in the soma may be perhaps driving the extra divisions leading to the multilayering phenotype. But what I was most interested in is whether this phenotype is specifically due to the loss of 5B, perhaps due to the loss of an RPS5B containing specialized ribosome, which might be translating specific factors required for oogenesis, or perhaps whether this is due to a defect in ribosomes and translation more generally. So to do this, I wanted to knock down some canonical ribosomal proteins in this region where RPS5B is the predominant RPS5 paralog. So this driver that I've used, you can see it's only in the germline, and it also excludes the germline stem cells, but otherwise mimics the RPS5B expression pattern quite well. So by knocking down other ribosomal proteins which don't have any paralogs, you can see, firstly, there's a lot of TOR activation. Also, there's no eggs are ever produced. These egg chambers die before getting to that stage. If we zoom in closer, you can see again that, yeah, we recapitulate the somatic multilayering phenotype. And also, despite only driving this RNAi in the germline, we can see somatic TOR activation, including in the follicle stem cells, suggesting that this is being driven by the germline TOR activation, which fulfills the germline to soma signal. So all of this together suggests that this phenotype we see when we lose RPS5B is actually due to a loss of ribosomes more generally. But to test that a bit further, I wanted to also knock down some proteins involved in ribosome biogenesis. So UDD interacts with TAF1B and TAF1C-like to regulate RNA pol1 transcription to transcribe the polycistronic pre-RNA. And UTP6 is involved in cleaving that pre-RNA. So these are both pretty early steps in ribosome biogenesis. And again, you can see that mutating UDD or knocking down UTP6 causes this ectopic TOR activation, causes egg chambers to die way before making any eggs. And once again, causes the multilayering and activation in the soma, including the follicle stem cells. So from all of this together, I believe that this phenotype that we are seeing is due to ribosome biogenesis stress rather than due to a specific stress caused by the loss of RPS5B translating specific products. So just to quickly conclude, most ribosomal protein paralogs in Drosophila and Monogaster are not required for viability or fertility. One of them is required for fertility, and that's RPS5B in the female germline. And I found that loss of RPS5B leads to this ectopic TOR activation in the germline and then in the soma, and that this causes egg chamber death at mid-oogenesis, resulting in full female sterility. However, this phenotype seems to be a more general response to ribosome biogenesis stress rather than to the loss of a specialized function of RPS5B. And I guess we've also realized that when investigating whether a phenotype is due to specialized ribosomes or perhaps an extra ribosomal function, it's very important to consider the different expression patterns and levels of these paralogues. So thank you to the organizers for inviting me, and also thank you to my brilliant lab, especially to my supervisor, Philippe, and to the Cambridge Fly facility for lots of injecting for all of my mutants. Thank you also to everyone that shared reagents and ideas with us throughout my PhD so far. And thank you guys for listening.
Perfect. Thank you so much, Katy. That was really, really good. Okay, so just a quick reminder that we'll be answering any questions for Katy at the end. So that just leads me to introduce our next speaker, who is Dr. Shajahan Anver from University College London. Shajahan obtained his PhD from the University of California, Davis in molecular biology. After working as a Max Planck Fellow at the Max Planck Institute in Cologne for 2 years, Shajahan then moved on to work as a research associate at the Institute of Healthy Aging in the Department of Genetics, Evolution and Environment at University College London until present. So today, Shajahan will be discussing his work on how aging-associated long non-coding RNA1 or AL1 slows growth and prolongs chronicle lifespan by inhibiting protein translation in fission yeast. So over to you. I can see everything.
Yeah, thank you, Carl. So thank you very much for the Biochemical Society for giving me the opportunity to present my work. So today I'm going to talk to you about a long non-coding RNA at the crossroads of cellular aging and protein translation. So I'm Shajahan Anver from both BALA and ALEC groups from the UCL Institute of Healthy Aging. Let me first introduce why we are interested in the long non-coding RNAs. So at the advent of RNA-seq technologies, so we now know that there are more long non-coding RNA genes than the protein coding genes. For example, in the human genome, there are about 20,000 protein coding genes, but the long non-coding RNA genes are about 28,000, and they've been discovered every day. So those of you who are not familiar with what long non-coding RNAs are, so very broadly, long non-coding RNAs are any RNA which is greater than 200 nucleotides. So this is an arbitrary cutoff based on RNA-seq techniques. Like mRNAs, these long non-coding RNAs are transcribed by RNA pol II, and like mRNAs, they are capped and polyadenylated, but unlike mRNAs, they are very, very lowly expressed. So why are we interested in long non-coding RNAs in terms of aging or healthy aging? So we know that there are many long non-coding RNAs they are working on many pathways of molecular hallmarks of aging, including epigenetic regulation, proteostasis to cell proliferation, as you can see these many examples which have been shown in this figure. So these have been shown in either human studies or else in mostly mammalian model organism studies. So as yeast biologists, why are we interested in these? So what does the yeast play? So we have observed that in the yeast, there are many long non-coding RNAs highly upregulated during cellular aging in Schistosaccharomyces bomby or fission yeast, especially during many stress conditions. For example, during meiosis or during aging, at the onset of aging, where the 100% of the cell population is alive at the stationary phase, or 50% of the population is already dead. Or else also we have observed in quiescent cells after 24 hours of nitrogen starvation or even after 1 week, we have seen many long non-coding RNAs get upregulated compared to the mRNAs. So one quite obvious question come to our mind that during aging or during stress, quite obviously that the regulation at the chromatin level has gone off or went down. So many of them could be simple biological noise. But we were interested in asking the question that at least some of them could be biological signal. So that's what we went on to ask. And then we deleted and overexpressed more than 100 long non-coding RNAs in fission yeast and screened them for effects on cellular longevity. In that expedition, we have identified one long non-coding RNA, which we named later on as aging-associated long non-coding RNA, or AAL1, which the overexpression leads to lifespan extension. So as you can see, we expressed within a thymine repressible NMT1 promoter. So this extends the lifespan. And then when you add thymine to the NMT1 promoter to the cells, when the expression is repressed, then this phenotype is gone, confirming that this longevity or long lifespan is due to AL1 overexpression compared to an empty vector control. We went on to delete AL1 with a CRISPR-Cas9 system with two different sgRNAs or guide RNAs. We found that deleting the AL1 really reduced the lifespan or shortened the lifespan compared to a wild type. So unfortunately, we don't have any functional information for AL1. So as obviously as a yeast lab, we turned to genetics to find out what AL1 does and how it really extend lifespan. So in order to do that, we combine the AL1 deletion mutant with a library of 3,420 gene mutants. So these are non-essential genes, meaning that the deletion of them doesn't cause lethality. So we made all possible double mutants and queried them for genetic interactions or defects in growth. So when we did this analysis, we identified significantly genetically interacting proteins with AL1, and then these genes were mostly enriched for translation or ribosome biogenesis. We did a similar experiment with similar screening with AL1 overexpression lines, and then we found these genetic interactions were stronger, similar to the phenotypes we observed before, and also it revealed similar functional enrichments. Encouraged by this, so we asked the question, if you overexpress AL1, does that affect the transcription of any transcripts? So we did an RNA-seq analysis in the AL1 overexpression, and during the exponential phase or the growth phase, we didn't find that many genes were affected. But during aging, 7 days after the chronological lifespan, we found that many genes which are involved in promoting lifespan were upregulated. But most interestingly, genes which are involved in cytoplasmic translation and ribosome biogenesis were downregulated. So this further confirms our suspicion that AL1 might be involved in either translation or ribosome biogenesis. Now, naturally, we were curious now, where does AL1 localize to? So in order to answer that, we turned to small molecule RNA fish. So with a line which overexpresses AL1 in the chromosome locus, so we can see the nascent transcript. So the DAPI stain is shown in blue here, and the AL1 transcripts are shown in green. And then when we looked at the localization, we found that mostly AL1 is localized in the cytoplasm, although we see the nascent transcript in the nucleus. So this further confirms that most potentially that AL1 is involved with the translation and promoting lifespan through moderating translation. So to understand how the AL1 functions is we have to know that what proteins AL1 is interacting with. By knowing the AL1 interacting ribosomal proteins or AL1 binding ribosomal proteins, we will be able to tell how AL1 really moderating the translation. So in order to answer that, we pulled down AL1 interacting proteins, and we found a similar enrichment of cytoplasmic translation proteins were pulled down with AL1. And when we did a string analysis for all known physical interactions of these pull-down proteins, we found that many of them were either ribosomal proteins or ribosome-associated proteins. And other than that, we also found there were many metabolic enzymes as part of these pull-down proteins. So now we know that in different systems, including mammals, that many metabolic enzymes function as ribosome-associated proteins or RAPs. So what we concluded from this finding is that most potentially that AL1 really physically associate with ribosomes. So in order to answer that question, we turn to polysome profiling. So that's the gold standard to answer that question. So how polysome profiling works, if you're not familiar with, is that you use the sucrose gradient to separate your lysate into different gradients, and you send it through a gradient fractionator. Through the gradient fractionator, you can collect each fraction and extract RNA and ask the question, where does AL1 or any other RNA which you are interested in is localized to, in which fraction and how much enriched it is. And also, other than that, you can also measure the amount of free ribosomes or monosomes or polysomes in each strain. So when we did that experiment, we found that AL1 is mostly localized in the free ribosomes and in the monosome fraction, but also there's a decent amount of localization found in the polysome fraction as well. This is very evident when you look at the control, a PPB1 mRNA, which is a protein coding gene, which is lowly expressed. That's what we use this control, because this kind of match well with a long non-coding RNA, which is lowly expressed. So this protein coding gene transcript is mostly localized in the polysome as expected. So this further confirms that the AL1 indeed physically localized to the ribosomes. Using these polysome fractionation profiles, we also observed that in these profiles that when you overexpress AL1, the amount of ribosomes are reduced, whereas when you delete AL1, the amount of ribosome content increases in the cells. So this is what I'm showing here is during the growth phase or the exponential growth phase. We did similar experiments for the onset of aging or the stationary phase, and also during the aging process or the chronological lifespan. Then we found the pattern is similar. So we wanted to quantify this observation. So we went on to calculate polysome to monosome ratio, which is the translation rate, or we also calculated polysome and monosome together, or the relative ribosome content relative to their respective controls, which would really tell us about the translation capacity. So when we calculated PM ratio, we found that though there were changes, but none of them were statistically significant in any time point. But when we looked at the relative ribosomal contents relative to their respective controls, we found that the ribosome content is reduced in the AL1 overexpressing strains and increased in the deletion strains during the growth phase, and it becomes very prominent during the onset of aging or the stationary phase, though we didn't see much of a difference in the chronological lifespan later in time. So this suggests to us that the translation, though the translation rates might not be much affected per se, but the total ribosome content is reduced in AL1 overexpressing cells. So the quite obvious questions come to our mind that ribosome content and growth are very proportionately related and have a positive effect on each other. So if that is the case, if AL1 overexpression reduces ribosome content, then does it slow growth? It should be. So we looked at the growth rates of overexpressing cells. Indeed, they were really slow and the growth rates were reduced in the expression cells. So this is the similar system where we overexpress AL1 with an NMT1 promoter, which is repressed by thymine. So relative to an empty vector control, AL1 reduces growth, and when you add thymine to repress this promoter, and this phenotype is gone and rescued. So this suggests that indeed the reduction of ribosomal content has an effect both on the longevity and also on the growth. Now the final questions were, what are the implications of AL1 ribosome association on its molecular function? Among other questions, we also asked the question that, does the ribosome binding suggest that AL1 binds selected mRNAs and alters their translation? So in order to answer that, we pulled down the AL1 bound RNAs and sequenced them. And then we found the topmost AL1 associated mRNAs were ribosomal protein mRNAs. And relative to AL1 deleted cells, we found that both in the wild type and the AL1 overexpressing cells, these ribosomal protein transcripts were enriched. We also looked at our RNA-seq data to see whether the transcript levels were affected in the AL1 overexpressed cells. And indeed, we found that these transcripts were moderately downregulated. But these are not the only things. As you can see, all the ribosomal proteins which were downregulated were shown in the cyan. So there were many ribosomal proteins which get downregulated among all the downregulated genes which were shown in yellow. So among these ribosomal protein genes, RPL1901, which is a large ribosomal protein gene, or it's called RPL19 in other organisms, is an interesting one because in Cerevisiae, budding yeast, and also in worms, C. elegans, it has been known that reducing the transcript levels of RPL19 or deleting them extend lifespan. So we were excited by that. So we want to ask a similar question in fission yeast to see whether deleting RPL19 would have a lifespan effect. Then it would be able to explain what we see in the AL1. So when we deleted AL1, when we deleted RPL19, to our disappointment, that it really makes the cells very sick, and the large subunits disappear, and also they die very quickly and live very short life. So we were a bit disappointed, but thinking about it, we realized that AL1 is a long non-coding RNA, and it really reduces RPL19 to very moderate levels. So what we went on to do finally is we express RPL19 with an NMT1 promoter in its native locus, and we reduced it with a low amount of thymine, repressed the transcript with 3 micromolar, which represses the transcript for about 2 to 3 fold reduction. Then when we found that if we reduce it moderately, then we see a moderate lifespan extension in fission yeast. So this confirms that AL1 somehow moderate the transcript levels, or maybe the protein levels of RPL19, and thereby extend lifespan in fission yeast cells. To summarize what I have told you so far is during the growth phase, AL1 is repressed, so we can't detect AL1 during the growth phase. But in turn, if we overexpress AL1, it slows growth, makes sense. And during aging, AL1 get upregulated, and in turn, if you overexpress AL1, it prolongs longevity. Whereas when you delete AL1, it shortens lifespan. I have also shown you that AL1 is mostly localized in the cytoplasm, and it goes to the ribosomes. It might be doing ribosome stalling, but we were unable to find any evidence to show that. But what we found was AL1 binds to selected ribosomal protein gene RNAs and reduce their content. So thereby, we hypothesize that either this AL1 binding leads them for degradation and reduce the transcript levels, or else AL1 goes to the ribosome with these ribosomal protein gene transcripts and prevent them from translated. But either way, they are going to affect the stoichiometry of these ribosomal proteins, and as a result, will reduce the ribosome content when overexpressed, as we have seen in the overexpression line, as I have shown you before. Now, the final piece of evidence we are going to show that is ribosomes are highly related. And we know that both the ribosomes, the translation machinery, and the regulating proteins, all of them were highly conserved from yeast to humans. But the long non-coding RNAs are not very highly conserved across taxa. But the interesting thing about long non-coding RNAs is, though they were not conserved, their molecular mechanisms are conserved for many known long non-coding RNAs. So we were interested in asking the question, the AL1 has the potential to extend lifespan in higher organisms, so that with Nasif Alik's lab, and we expressed AL1 guts of female flies. So we used an UAS system, which is get induced by adding called RU. So when you induce AL1 in the midgut, we found that it extends lifespan compared to an uninduced state in Drosophila, suggesting that AL1, the function of AL1 or the molecular mechanism of AL1 is indeed conserved, giving us more to think about in the future. With that, I conclude my presentation and would like to thank the Yerkes lab, all the members, former and the current, for helping me in various ways, and the IHA community at UCL, and our collaborators, Sam Margaret lab, where Ziming Sun, a postdoc who performed localization experiments for AL1, and the Costas lab, Abu, a talented technician who performed mass spec analysis of our pull list, and in Nasif's lab, Ahmed, a talented PhD student on most of the fly experiments. And BBSRC and Wellcome Trust funded research. And finally, thank you very much for your attentive listening. And I would like to take any questions you might have. Thank you.
Yeah, perfect. Thank you very much, Shajahan. That was really, really interesting. Right, so now we can welcome some questions for any of the speakers. If you have a question, please type it in the box as shown, well, as shown you before. And again, please also state who your question is for. So, okay. So I think I'm just going to start with one quick question for both of you. So Katy, you know when you knocked out your RPS5B, do you see like a compensatory upregulation of RPS5A?
I haven't really tested that fully yet. I was thinking of doing, the problem is that ovaries are so very different from each other. I don't think I would want to do like Western blots or anything that used whole ovaries, because you're kind of comparing an ovary with lots of eggs to a slightly dying ovary. I could do IF clones.
Yeah.
Have a look. Yeah.
I would just be interested to see if you do have like an upregulation. That would be quite interesting. Shajahan, I just wanted to ask you, is there anything similar about the RNAs that the AAO1 kind of associates with? I know ribosomal proteins have like a top motif, and I wondered whether there was anything that was quite unique about them.
Not necessarily. So we don't know about motifs. We didn't do any experiments on that direction. So I think when it comes to RNAs, we try to find motifs, and it seems like AL1 has few motifs which would suggest that. So that some of these motif prediction programs even went ahead and asked, is that an snRNA? Or because it has some, these are poly A motifs, which could be really misleading, poly U motifs, which could be found accidentally in a long non-coding RNA, which is AT rich. So we were not really excited about going in that direction yet. But I think what we might have to do is to find the secondary structure of AL1, and that might be able to tell us maybe if there's any motif. I think.
So maybe some shape seek on the horizon.
Of course, yeah.
Okay, so we have a question that I think is for Katy. So what happens when you remove maternal contribution using clones?
I'm not sure that would. You can remove RPS5B and produce clones. They progress to about stage 8 of oogenesis, and then they collapse and die. You can't make RPS5A clones. It's lethal. Do you mean maternal contribution, like if you could make eggs without 5B maybe? Because we've been trying to think of how to do that, but I think that would need some kind of Degron system. There's no way to kind of get an egg without it currently.
Fair.
Yeah.
Okay. Any more questions for our speakers?
Can I ask one from Katy?
Oh yeah, go for it.
So if the defects in the germline development are due to, you assume that upregulation of the TOR pathway.
Yeah, it seems to be.
Okay, so have you kind of thought of, I don't know whether it's kind of like a sideline totally for what you are interested in, but adding rapamycin or something to inhibit the TOR pathway and see whether that really, because you looked at the phosphorylation of S6 to really indirectly measure the torque. So if you add something like rapamycin, will it really rescue these defects?
I've tried feeding rapamycin and the results have been inconsistent. Like sometimes it could partially rescue, like they progressed further through oogenesis and made some kind of malformed eggs. But then on some repeats, I couldn't reproduce that. So I'm not sure how much of that was due to, because I have to feed the flies the rapamycin, whether they're taking in different dosages. So now I'm just trying to like knock down, say, Tor and Raptor and do it genetically instead, because feeding was too inconsistent. But yeah, hopefully that will rescue it.
Shajahan, you mentioned ribosome stalling. Did you have a look at GCN2, GCN4, whether you see activation?
Yeah, I think, yes, disappointingly, we didn't find anything in the RNA-seq, or we also did some proteomics, which I didn't show here. So in none of them, we didn't find anything to suggest that. That was the first hypothesis we tested, to be honest. So we didn't find any quality control pathway genes or anything GCN or anything. Yeah, but I think we might have to do, in order to answer that, I think we might also have to do some riboseq to see whether there's anything to suggest that there's some ribosome stalling per se.
I was also intrigued to see that, in your polysome traces, AAL1 seem to associate mostly with the 40S small subunit. Do you have any, I guess, guesses or hypotheses as to why?
Yeah, so the other thing, like, it has been suggested by someone, which I kind of skipped, like when we look at the localization, someone believed that it could be really localized in the nucleolus rather than mostly. So we have a feeling that it could be that is highly possible, because there are so many, or I would say more than 5 or 6 nucleoli along non-coding RNAs known to function. And one of them has a very important function. When you delete, it is essential. So it is involved in liquid phase formation. And so I think it could be quite possible that it really comes with the 40S from there, and it involves in ribosome biogenesis. But unfortunately, none of the efforts we try to put in, and because the issue with fish, the smRNA fish, you can't do them in aging cells. It is very hard to do the fish. So we have to rely on this overexpression line, which is exponential growth phase. So it could be functionally less relevant somehow. So it's a little bit, some technical challenges we face. So yeah, I don't have an answer, sorry. Yeah.
Character building.
yeah, of course, yeah.
Sorry, Katie. I'll go.
Question?
Go on,
please.
The thing of reducing levels of RPL19, either did you do it, or did the worm and Cerevici people kind of reduce levels of any other ribosomal proteins?
Yeah, there were quite a few papers already, like, I don't know, 3-4 papers I know. They did genome-wide expression, genome-wide deletion. So these are deletion, I think they use deletion libraries, at least in the Cerevici, and they found quite a few in both replicative aging and then chronological lifespan to extend lifespan. So there, I think you can kind of easily find these papers. So there were RPL19, RPL18, RPS10, and also I think RPS6. these were all kind of genes which even overlap. Yeah, so yeah.
And I know yeast have quite a lot of paralogues. Do those genes, is there another RPL19, or is that ribosome really completely lacking L19?
It is. Yeah, no, I didn't really, because we did some experiments, but yeah, that will confuse, so that's why I didn't talk about. So we looked at both paralogues. So it is because of we have to rely on genetics because of this ambiguity in RNA-seq and proteomics. Because RPL19, all the genes I shown have paralogues. So that's what I tried both of them, and only one of them have extension. So it's that's what we think, that there could be functional specialization for RPL19A. So RPL1901 is RPL19A, and also fission yeast has a 19B. And we did some experiments on that line, and then so as I told you in the experiment, we found that when you delete, so unfortunately in cerevisiae they found deleting doesn't have an effect, serious effect, and extend lifespan. But in fission yeast they are very sick. You can't even revive them when you delete. And we looked at polysome fractionation and found that the large subunit is gone at the beginning until they grow to a certain, they recover, but at the beginning when you do polysome you don't even detect large ribosome subunits. So it suggests that they are not mutually exclusive, these paralogues. So yeah.
Cool.
Shajahan probably already said this, and I might have missed it, but I know long non-coding RNAs are quite poorly conserved. Is there any mammalian versions? But
no. I think so when it comes to long non-coding RNAs, I think they are conserved maybe in the mammalian clade, not down. Yeah. So
it's basically the pursuit of the elixir of life, potentially.
It's something analogous.
Yeah.
Okay, we have a question for Katy. So, what is your interpretation of the nuclear defects in the egg chamber? Sorry.
The nuclear defects, as in
the egg.
Okay. If you mean the nuclei of the nurse cells with the DAPI going super bright and then blobby, and I also see some nucleolar defects, but I think those late stage ones, that's just we're beginning to see the DNA fragment as the nurse cells die. If I'm misunderstanding your question and you mean maybe the kind of multi-layering of the follicle cells, which I mainly showed, I guess, through the multiple nuclei, then I basically think that there's extra divisions occurring kind of before a stage in mid-oogenesis, follicle cells switch from mitosis to endo-replicating. So I was interested for a while if maybe that switch was being delayed. But it seems like that's not the case. The defect is occurring before that stage. So I think there's probably some extra divisions happening before that, probably driven by this ectopic tour that I see in the soma. If that remotely answers what you're asking, I can't really tell.
I think so. Okay, well, actually, I'll just ask you one more question, Katy, because I'm going to ask you that annoying question that I know you get asked at every conference, and then I forget completely the answer. Have you, or are you, have you, or do you have plans to switch the promoters of the two, so RPS5A, 5B?
I'm not switching the promoters, I'm switching the actual coding sequences for A and B. Yeah, it's in progress. Haven't had much success yet, but I've adapted my CRISPR strategy slightly, and I'm actually kind of screening this week. So this will be the kind of key answer for whether or not the proteins themselves are functionally different, or if they're just different expression patterns.
You'll have to send me an email when you get those results.
I'll tell the whole world if I get it.
Okay, fab. So, that just leads me to kind of wrap up. I'd like to thank everyone for attending, and of course, thank you to our wonderful speakers. You can all continue the conversation online. You can follow Biochemsoc or PPP Publishing on Twitter. We also welcome suggestions for future topics and speakers to feature in the Biochemistry Focus webinar series. So if you have an idea for a webinar in 2023, we invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars, propose your webinar, and watch previous recordings at www.biochemistry.org. All of our upcoming webinars are listed on our website as well, and if you have missed any of the 70+ webinars that we've already run as part of the series or would like to catch them again, please visit our website or YouTube channel. So the recording of today's webinar will also be available to watch within the next couple of weeks. So please do join us on the 8th of September at 11 AM BST for our next webinar, which is titled How to Apply for Biochemical Society Grants, Education, Research and Outreach, where members of our reviewing panels will be providing advice on how to improve your chances of submitting a successful application. Finally, I just want to highlight that it's more important than ever to stay connected and engaged with your fellow researchers. So do join the Biochemical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, and I have taken really good advantage of these previously and they're really good to put on your CV. And you'll also get personal online access to 2 of their journals and a lot more benefits as well. So do visit the website to find out more. So again, thanks to both speakers and to all you for attending and the Biochemical Society staff who make all the magic happen behind the scenes. And I hope you enjoy the rest of your day.
Thank you.